Hello, and welcome to today's panel entitled Biopharma Panel Discussion, Inside Perspective from STAR Partners. I'm the moderator for this session today. I'm Dr. Tom Dubansky. I'm the president of Tempest Therapeutics, which is a clinical stage biotechnology company located in South San Francisco that is developing various therapies for oncology. The reason that I joined today in this panel, I am a board member of the CMTA, and also someone who's learning to live with CMT1A. And so I'm very excited today to talk to our panelists. And we have selected 4 different panelists who are the leaders of various companies, various biotech companies that are developing therapies for CMT. And today is an opportunity for us to understand from each of these leaders in the companies to tell us a little bit about why they chose to pursue CMT and exactly what they're doing in the context of developing new therapies for CMT. Certainly the community at large is well aware there are no available therapies that have been approved by FDA for treating the various subtypes of CMT. First, the panelists that will be joining us today are Dr. Mark Forman, who is the Chief Medical Officer of Passage Bio, Dr. Suyash Prasad, who is the Chief Medical Officer and Head of R&D at Taysha Gene Therapies, and then Dr. Shoshana Schendelman, who is the CEO and founder of Applied Therapeutics. Unfortunately, Dr. David Horn, who is the CEO of Farnext, was feeling under the weather today. And at the end of this panel discussion, I'll just say a few words about Farnext for those in the community that are unaware of this company and what they're doing. So with that, during this session, as I said, we're just going to find out a little bit more of these about each of these companies from the representatives that are here today. So let's start with Dr. Mark Forman. And again, I'd like to thank you and each of you for agreeing to join the session today. And would you please provide us just with a general background of Passage Bio and the mission, and describe the approach that you're taking to develop a therapy ultimately to be evaluated in patients with CMT.
Yeah, well, thanks, Tom, and thanks for the opportunity. Thanks to the CMTA for the invitation to participate today. It's really quite an honor. I'm the CMO at Passage Bio, which is a genetics medicines company based in Philadelphia that's developing therapeutics for diseases of the central nervous system. We're formed from a research collaboration with the University of Pennsylvania and Jim Wilson's group. And in fact, for those who participated in the last one of the breakout sessions just previously, you heard Jim talked about the CMT2A program that is a partnership with that we are developing in partnership with them. Passage has a pipeline of gene therapies focused on both rare and now common CNS diseases in both adults and pediatrics. So we're focused on just quickly lysosomal storage diseases, neurodegenerative diseases, including adult diseases like ALS and Alzheimer's disease, epilepsy, and obviously for this discussion, Charcot-Marie-Tooth disease is something that we're actively working in. So that's a little bit about Passage. So Tom, the other question was can you remind me what the second question?
Well, if you could just talk about a little bit why Passage decided to pursue CMT, and particularly you mentioned that you're targeting CMT2A.
Yeah.
And if you could just tell us a little bit about your approach.
Yeah. So we are a gene therapy based company, and as we're using the adeno-associated virus as a means of introducing genes into the brain, and so so one of the reasons that we're excited about this is one of the reasons that CMT is a great target. We think is a great target for this approach is because the virus, when we transfects or expresses the proteins that we're introducing at very high levels in the relevant cells in the spinal cord, both the motor neurons and the sensory neurons. So we're actually getting the gene to the right place, which is what we're trying to do here. CMT2A in particular, which is what we're targeting, is a disease of the neurons themselves. So it's, again, we're getting the gene into the right cell type. And in addition, on top of that, the genetics of CMT2A are well defined. So it's predominantly an autosomal dominant disorder. So the approach we're taking is is actually a combination approach, which is we're both trying to knock down the mutant protein. So by using the virus, we're trying to get it into cells and actually reduce the expression of the abnormal protein, and then increase expression, and then express the normal protein to compensate for that. So it's a combination approach that we're using with the virus, and again, CMT, because of the 2A, because of the gene and the known genetics with mutations in the mitofusin gene, are really a really nice target, because of we should be able to get the virus into the relevant cells. And if those who listened to Jim Wilson's talk just at the prior breakout session, he showed data in animals showing that approach is working, at least in a mouse model of CMT2A.
Well, that's very exciting. Can you please provide the community with just kind of a sense of, where you are in the development of this exciting therapy, and have you already advanced to testing in, for example, animal models of CMT?
Right. So in fact, that's exactly where we are. We're testing we've done proof of principle, what we call proof of principle studies. If we introduce the gene in an animal model, in this case a mouse model, and we're also working towards larger animal species, testing that, and if that is successful, we would then in parallel identify. Again, this is work done in collaboration with the group at Penn. We would then identify the best construct, the gene therapy, that we would then take through the necessary safety studies and pharmacology studies that would allow us to then bring something into the clinic. But we're still in the proof of principle stage, and the preliminary data, like I said, Jim showed some very nice data earlier, just a little while ago, that showed that it seems to be working. We need to continue that, pursuing those preclinical studies. But if everything is looking promising, then the next step would be taking that through the necessary preclinical work to support advancing into the clinic.
Terrific. Well, thanks. Well, we'll come back to you later in the session. So let's move on to the other speakers and understand what their exciting developments are as well. So again, very thankful that Dr. Shoshana Schendelman could join this session today, and she is the CEO and founder of Applied Therapeutics. So welcome, Dr. Schendelman, and can you please let the CMT community know what it is specifically that Applied Therapeutics is doing in terms of developing novel therapies for CMT, and some of the reasons that led you to guide your company into pursuing this disease, and look forward to hearing about it. Thank you.
Absolutely. Our story is a little bit different, but we've been targeting an enzyme called aldose reductase, which is very well understood to cause many diseases. And in fact, we had developed a very specific drug, AT007, which inhibits the aldose reductase enzyme and is also CNS penetrant. For all the reasons that Mark just spoke about are relevant in CNS diseases. And specifically, aldose reductase had been previously implicated in another rare CNS disease called galactosemia. And so we were developing this drug for galactosemia, and we're pretty far along in the process. So we had already completed a pivotal study in adults with galactosemia, and we had just started a phase 3 pediatric study in children aged 2 to 17 with galactosemia. And at that point, there was a really important research discovery in the CMT community. And I think that this is a really great story and sort of illustration of why research in general is so important. So I think, as a lot of people know, many patients with CMT have an unknown genetic cause of disease. So there are many patients, for example, with CMT2 unknown. They're diagnosed as CMT2 based on symptomatic presentation of disease, but we don't really know what the genetic cause is or the pathogenic or molecular pieces that lead to the symptoms that they have. And all partners in the community of research. So it was a big collaboration between the CMTA, Hereditary Neuropathy Foundation, Inherited Neuropathy Consortium, have been working on identifying some of these genetic or molecular causes of disease for patients who really don't know why they have CMT2. And out of that research initiative came an important discovery that about 10% of patients with CMT2 have disease because they have a genetic defect in the SORD gene. And so this is now called SORD deficiency. And what was really amazing is that as soon as we knew what the genetic cause of that subset of disease was, we knew exactly why they had neuropathy and a motor neuron disease, because that enzyme is really well understood. So it turns out that the SORD enzyme is the enzyme that follows aldose reductase. So aldose reductase, the enzyme that we had already been working on blocking for a few years, is responsible for converting glucose to sorbitol. And normally, the enzyme sorbitol dehydrogenase, or SORD, then converts sorbitol to fructose, and it's used for energy production. So patients that are missing the SORD enzyme can't metabolize sorbitol. And aldose reductase converts glucose to sorbitol, and it has nowhere to go. And it builds up to very high levels in patients with SORD deficiency. So it was an amazing finding of why these patients are ill. We've known for a long time that sorbitol is toxic to many cells, and it's especially toxic to neurons. So understanding that these patients are missing the enzyme to metabolize sorbitol, that they have very high sorbitol levels, explained all of their symptoms of disease as well. But even better than that, we had a drug to block production of sorbitol. So this was an opportunity for us to offer a pretty late stage drug to a newly defined disease. So even though SORD deficiency has only been identified for about a year and a half, we're moving into a registrational trial for patients with SORD deficiency this year. We were able to do that because our drug was so far along for another rare disease, but also because there had been so much research for decades on the aldose reductase and sorbitol dehydrogenase pathway. So it's a great sort of plug for why research in general is important, even if we don't know at that moment when it will be used. It does allow us to put pieces together very quickly when something comes up, like the identification of SORD deficiency as a cause of disease. So we were really fortunate that all of these pieces came together, and we were even more fortunate that the CMT community collaborates so well. That advocacy organizations like CMTA, and physicians who are all united through groups like the Inherited Neuropathy Consortium were able to just come immediately and say, we have a drug for this. We'd like to, develop it for SORD deficiency, and how can we work together? And it allowed us to move quite quickly. So we just completed a pilot study in patients with SORD deficiency this summer. We announced our results a couple of weeks ago. And it's, not a big surprise. Our drug AT007 blocked sorbitol production in patients with SOR deficiency, and we're able to reduce sorbitol levels pretty substantially by about 66%. But that was just a pilot study. So now we're planning our large study. We're set up to start that towards the end of this year. And we're very excited about the potential of bringing a drug to this disease in such a short period of time.
Well, my goodness, that is just a wonderful story, Dr. Schendelman. It's nice to hear. It seems like some of the early partnership that you had with the CMTA and the HNF really sort of facilitated your work in the understanding of the mechanistic reasons why it made sense to test AT007 in this clinical setting. And it sounds like that you will begin to enroll, I believe you said later this or early next year for your approval study in CMTA.
Yep, it'll be later this year. We just announced the launch of it ahead of this trial. So it's called the INSPIRE clinical trial. And it's in patients with SOR deficiency, age 16 and older. And we'll begin enrolling that before the end of this year.
Well, that's so exciting. And maybe I'll just ask you one further question before we move on to Dr. Prasad. But just given that the size of the number of patients afflicted with CMT2A is relatively small, is this a single arm study, or is it, if you can tell us a little bit about how that approval study is set up?
Yeah, absolutely. So it turns out that SOR deficiency is one of the largest causes of hereditary neuropathy, or CMT2. So about 10% of the CMT2 population has SOR deficiency. So that means there's about 3,300 patients in the US and about 3,500 in Europe. So this clinical trial will have sites in the US as well as Europe. And I know it is a rare disease, but that's quite a number of patients. What we do need to think about, though, is because it's only been identified as a unique disease for about a year and a half, the number of patients who know that they have SOR deficiency is not 3,300 in the US. So we do have quite a bolus of patients. We've had a lot of demand to get into our pilot study and then also to get into this registrational trial, which we really appreciate and I think speaks volumes about the patient community and their willingness to participate in research. So to all of those patients who have contacted us, we really thank you. This should not be difficult to fully enroll our trial. But Tom, to your question, it'll be about 50 patients total in the US and Europe. There will be a placebo arm, but it will be a 2-to-1 randomization of active versus placebo. So your chances of getting the active drug are twice as high as getting placebo. And then as soon as the study meets its endpoints, of course, the placebo group will have an opportunity to cross over to active in an open label setting. So I'm sure there's been a lot of discussion within the community about the importance and the need for trials to be placebo controlled, even though everyone would like to get the active drug. So we've worked very hard with FDA to design this study in a way that provides a clear result, but also provides benefit, hopefully, to all of the patients who participate.
Well, thanks so much for summarizing that very exciting trial that Applied is now engaged in, and certainly it's a bit redundant, because I said it at the top of the session, but all of us living with CMT know that there are no approved therapies, and so we're so thankful to all of the various pharma groups that are developing new drugs for CMT. And with that, why don't we move on to our next panelist, which is Dr. Suyash Prasad, who is the Chief Medical Officer of Taysha. And just a few questions related to what your activities are at Taysha, but another emerging biotech company engaged in gene therapy that we've been reading about in the last year. And Dr. Prasad, could you just please help the community understand what it is that Taysha is doing why you chose to target CMT as well, and help us understand better what the company is engaged in.
Absolutely. So thank you for the question, Tom. Thank you to the CMT Association for the invite, Laurel in particular, and it's been nice to hear from Mark and Shoshana thus far. Taysha Gene Therapies, we are a gene therapy company, fairly new. We've been around for about 18 months or so. Our technologies are based on a foundational partnership with the University of Texas Southwestern under the leadership of Dr. Burj Manassian, who's the chair of pediatric neurology, and Stephen Gray, who is a highly regarded gene therapy scientist and directs the gene therapy vector core at UTSW. Our focus is on using AAV gene therapies to treat rare and severe neurological disease. Many of us at the company have a lot of expertise in gene therapy and have a lot of experience in treating rare, severe neurological diseases. My background is I'm a pediatric intensivist by training, so very used to treating children with severe metabolic neurological diseases, and have spent a number of years in gene therapy and rare disease drug development research. We have 27 programs, and they spread the whole range of drug development. We've got a handful of programs in very early stage development, so just at the stage where we've just identified a construct, designing a construct, and thinking about proving principle in clinical trials. We've got programs in the IND-enabling preclinical animal study phase, and we have programs actually in clinical trials currently also. I think specifically what's relevant for this organization are two of our programs, one which is in a very early phase and one which is in a very late phase. So the program that's in the very early phase is the gene therapy approach for CMT4A. Obviously, I'm sure many of you know that this is a mutation in the GDAP1 gene, and in partnership with Stephen Gray at the University of Texas Southwestern, we've been able to identify the gene that's been missing and replace it by wrapping up the DNA that codes for that particular protein within an AAV9 capsid. And we're just going through the late construct design, early proof of principle phase in the animal studies. So that's pretty early on. The other program that's very relevant to this group is our program in giant axonal neuropathy, which is actually a later stage program. This is a program that's already in clinical trials. It has, we've dosed 14 patients. It's the first ever gene therapy study to dose a gene therapy intrathecally, i.e. you insert a needle into the base of the spine, infuse the drug that way. You have the patient lie flat, tilted head down, and allow the gene therapy to bathe the brain and the spinal cord. And we've dosed 14 patients at 4 different doses. And in the data that's been shared, the clinical data that's been shared thus far, the medium low and the medium high dose, we've had some really stunning clinical results in that the disease progression halts absolutely. And I think the reason it's of relevance to this group here is that the underlying disease of giant axonal neuropathy is due to a mutation in the protein gigaxinin. Gigaxinin is a protein that is supposed to break down waste, old, denatured structural proteins within the neurons that should be recycled, but they don't get recycled because there's an absence of gigaxinin. So in the absence of gigaxinin, the protein, you get a buildup of these waste, abnormal structural proteins in the axons of the neurons. The neurons swell, they start degenerating and becoming inflamed, and then the electrochemical transmission down the neuron is compromised, which then results in the clinical phenotype of giant axonal neuropathy. Now, by replacing the gigaxinin gene, we can demonstrate recovery of that nerve and therefore an amelioration of the ongoing progression of this disease. The reason it's of critical relevance to this organization here is that there's a large group of patients. In recent years, it's clear that there's a large group of patients who've been diagnosed with CMT type 2, so the axonal forms of CMT, that may actually have a mutation in the gigaxinin gene. which is why it will be important to if you haven't had a genetic test and you've got the clinical diagnosis of CMT type 2, ask your physician to get a genetic test, because it could well be, if you're one of the 1 to 6% of patients, which is what the literature suggests, that actually has a mutation in the gigaxonin gene, you may well be amenable for this gene therapy approach that's currently in late stage clinical trials.
Well, that's very exciting, because one of the questions that I was very anxious to ask you is that, of course, Taysha recently announced that there was a collaboration between the HNF and the CMTA to administer genetic tests in particular patients, and so I think this is exactly exactly what you're summarizing right now.
Exactly, and I think that the comment I would make is that we now have what looks like a treatment for giant axonal neuropathy, gene therapy. It's not licensed yet. Sure. We have some. We will be talking with the regulators in the US, Europe, and the UK over the next 3 months or so to talk about a pathway to getting a regulatory approval for this really trailblazing gene therapy approach. And I anticipate that will be successful in the short to medium term future, and therefore we will have a gene therapy treatment for patients who have a mutation in the gigaxonin gene. And what's clear is that there are many patients out there who've not been diagnosed with this. They have the symptoms, they may be mischaracterized with a form of Charcot-Marie-Tooth, but we want to offer the opportunity, and we've actually just started a partnership with a group called GeneDx who do genetic testing for a whole host of different genetic mutations in neuromuscular disease. And it's in the process of being set up and rolled out over the next 3 to 6 months. And what it will mean is that the clinicians that treat many of the patients and families in your audience will be able to send off a test of a particular patient with what they suspect may be giant axonal neuropathy, send it to GeneDx, and there'll be no insurance cost or no cost to the patient or the clinician. It'll be covered by Taysha. And that way we anticipate diagnosing a number of patients who have a mutation in the GAN gene that are currently undetected at the moment, who then may be amenable to our gene therapy approach.
That's just such an exciting development, Dr. Prasad. And just to clarify for the CMT community, I'm guessing that this genetic test is performed with just a blood sample.
It's a simple blood test that tests for a number of common mutations that cause axonal neuropathies, and GAN, giant axonal neuropathy, is one of them. Yes.
Well, that's really very helpful, and thank you. I can say in my own experience, getting to a definitive diagnosis of 1A, which, as you well know, is a relatively common form of CMT. It was, frankly, a long and somewhat scary process, and in my primary care physician really hadn't heard of CMT since he was in medical school, and didn't have any patients actually that had the disease. So certainly your activities in creating awareness in the community, as well as what the other agencies like CMTA are doing, is really very exciting indeed. So thanks very much for that. So let's return to Dr. Foreman, and we've heard today in just this first part of the panel discussion that there are gene therapies which give the promise of having a cure for CMT. That's what's so exciting about it. And then we heard from Dr. Schendelman, who through some part of serendipity in thinking about the mechanism of action of the drug that she was developing, how it ultimately related to CMT, that is a therapy. I'm just wondering, Dr. Foreman, if you could maybe just give us a little bit of a perspective, sort of the benefits, or just so that community appreciates the difference between a curative therapy and a therapy that can be used to treat symptomatology. And how do you see in the future, these two alternatives to treating CMT evolving?
Yeah, so that's an important question. I mean, I think that there is in the field, there's a need for, for both, right? You need to I mean, there's an, the gene therapy will probably be best, the populations that will benefit the most are the people who are going to be very earliest in the disease process, because once you start having, damage to the neurons, as many of you know, the neurons don't grow back. So once the damage that happens is generally is thought to be permanent. And so you're going to need to have both symptomatic approaches, which manage the symptoms that any individual subject has, but as well as what we call disease modifying approaches that actually get at the core root of the disease. And, so the gene therapy is really focused on the latter, trying to correct the underlying problem with the hope that will then prevent, stop the disease in its track. Now, there are some examples, and I mean, the results that Siyash just talked about are really exciting in terms of this genetic approach to giant, to the giant cell axonopathy. But we're now, we're doing this approach across a range of diseases. But the importance for us is to try to, and that gets to the point that you also heard talked about, was diagnosing these people and the genetic mutations, because the people who probably may benefit the most are the people at the earliest stages of the diseases. So you need symptomatic people, we definitely need symptomatic treatments for the people with active disease, but we also need, continuing to work on disease modifying approaches for everyone as well. So they go hand in hand in my mind.
Yeah, that's really very helpful. And in general, when Passage Bio decided to develop therapies for CMT, what were some of the important aspects of the disease that led you to think that this made a lot of sense? I mean, there's, of course, for many CMT types, the damaged gene is already understood, and I think you already addressed that. But people that have been living with CMT for so long, and there's been some therapies, some of which continue to be in development, but with no approved drugs. So as for example, the various patient advocacy groups like CMTA, has that been helpful in the context of refining the strategy at Passage Bio?
Yeah, well, I mean, for all of our programs, working closely with the advocacy groups is critical when you're working in these rare diseases. I mean, I think I think you've heard all of us talk about that. I mean, I have some experience in sort of, in terms of common diseases, let's say Alzheimer's disease, and you don't, it's just that, it's very easy to, to identify the individuals that are affected. When you're working in the rare diseases, you need to do these in partnerships with the patient advocacy groups in order to really advance the programs. Some of that is like what you heard about, sponsored genetic testing programs. But it's also those research collaborations, getting us in touch with the experts. One of the things that the CMTA has been involved with that's going to be immensely helpful for the CMT2A program that we're working on is these natural history studies that you have sponsored, which allow us to really understand the progression of the disease, so that we have a reference population that we can see how our treatment is working compared to people to the others that are affected, because it's very difficult sometimes to do what we call controlled studies where you have placebo groups. So we heard Shoshana talk about that, but for a lot of our for these rare populations, we do open label studies where everybody gets the active treatment. And then we need these natural history studies in order to provide a relative comparator. So that work is immensely helpful for the field. And so we can't do any of this without the advocacy group. So this is really a partnership in research, and it needs to be bi-directional, where we're learning from the patients, from the caregivers, from the medical experts, and we're sort of trying to make sure that they understand what we're doing. So it's a really two bi-directional communication that's critical for the success.
Well, that's just wonderful to hear that, and thank you for those points. Again, speaking as someone that has CMT and is concerned about my family members and my interaction with the CMT community over the last 5 years, it's so clear to me that you have a patient population that's just frankly aching for new therapies. And And I think, I hope that the community is aware today that both Taysha and Passage Bio are led by two stalwarts in the gene therapy field, of course, with Jim Wilson and Stephen Gray, as already mentioned. So I'd love to ask a couple more questions with Dr. Schendelman, and at least for me, and I should have done a little bit more of my homework. It's so exciting that you're already engaged in an approval study with 50 patients. Can you give some sense to the community when you think that trial will be enrolled and when we can hope to start hearing the early data? It was just exciting that you're already seeing a biomarker response in sort of your early phase clinical development in CMT, but just very curious, of course, where you think that you could begin to start seeing some real results.
Yeah, we're very excited about it. So, as with a lot of things, we can't predict exactly, but I'll try to give you an idea. It will depend on how quickly we can enroll the trial, as you suggested. So I think there's two parts to that. On the one hand, we need people to participate in the trial, which I think we're in good shape with, given the turnout so far in the community. We'll, of course, screen everyone and get as many patients who qualify as possible enrolled. We've designed our trial as a two-part study, and this is a very similar approach to what we've done in galactosemia, where we were also looking at a biomarker reduction as well as long-term functional outcomes. So the SORD deficiency study is designed the same way, to look at reduction in sorbitol over a few months period of time. And then looking at long-term functional outcomes, which will probably take a few years to show an impact on. So what that allows us to do is to hopefully show an impact on the biomarker, which here is sorbitol reduction, and to apply to the FDA for accelerated approval based on the biomarker while the long-term functional outcomes are ongoing. So there's, many detailed things that have to happen there. I won't bore the community with it. We're working, as quick as we can to get all of that work done. But I think in terms of the biomarker outcome, we're reasonably looking at something within a year potentially of starting the trial. And that's what accelerated approval is designed to do for diseases where there are no treatments available. So that is true for SORD deficiency. And where there's a biomarker that's reasonably likely to predict clinical benefit. So we know that sorbitol is toxic, and that's what's causing the neuronal dysfunction, and we can reduce sorbitol significantly to permit availability of the drug or accelerated approval of the drug while the long-term functional outcomes are studied, which can take a few years. So that's the approach that we'll be taking here. And I think that it's been really great to see the level of sorbitol reduction already in the pilot study. That gives us some confidence moving into the registrational trial. I think it is reasonable to expect that we can see a similar level of reduction in the larger study as well. And I see a couple of questions popping up here. So if it would be okay, Tom, is it okay for me to just address a couple of those biomarker questions?
Absolutely. In fact, the next thing that I was going to do was to ask you and Dr. Prasad and Dr. Foreman to really say anything else that hasn't been asked that you think is important to this forum.
Sure. I'll be really quick with mine. I'm seeing a couple of the same questions coming up in the Q&A box here. For SORD deficiency, there's no downside to blocking sorbitol production. So in healthy individuals, it's a very small amount of your glucose that's metabolized into sorbitol. But in patients with SORD deficiency, that very small amount, it's only about 1% of your glucose that's metabolized to sorbitol. It's not the primary way that your body generates energy. But in patients who can't break down sorbitol, even just the 1% production of sorbitol from glucose accumulates into a really large amount. So patients with SORD deficiency have about 100 times higher levels of sorbitol in their blood and tissues than unaffected individuals. So I see this question popping up of, well, what happens? What's the downside of blocking sorbitol production? And there is not a downside, because if you block the aldose reductase enzyme, glucose is just metabolized through a different pathway, through your normal metabolic pathways where the other 99% of your glucose goes. So, and we know this from the other programs where we've studied aldose reductase inhibition as well. If you block aldose reductase, there's not anything bad that happens upstream or downstream. It's one of these funny enzymes that we have, but it doesn't do all that much. In a good way. It can cause problems, but there's not a downside to blocking it. So we don't see glucose levels go up when we block sorbitol production. So maybe that was a little more detail than anyone wanted to know about this, but I saw the question popping up. So thanks, Tom, for letting me answer that.
No, it's really very helpful and very clear that you seem to have already an existing fairly large patient safety profile with this drug. So putting it into this new indication, meaning CMT, is really already have laid a lot of groundwork with this really very exciting drug. So thank you for that. Dr. Prasad, same question for you. Is there anything that hasn't been discussed in this forum that you would like to talk to us about Taysha? I mean, it's really terrific news that you just helped us understand is actually, how these gene therapies are applied. And one thing I want to make sure is really very clear to the CMT community, both Passage Bio and Taysha are already engaged in clinical studies, not necessarily on CMT, but potentially, as you already indicated. And I'm sure the community is well aware that these are therapies that have been developed for many years, but now are the subject of approved drugs. But it takes quite a bit to go from curing a mouse to going through the safety studies and manufacturing these very complex therapies. And we, of course, as a community, are so delighted that this is exactly what you're doing. But can you tell us anything more about Taysha that we should know?
Sure. I think I'll make three brief comments, one of which will address one of the questions in the chat. The first is that we have this large suite of programs, 27 programs, as I mentioned, all focused on AAV9 gene therapies for treating rare and severe neurological diseases. And part of the value of having such a broad platform is you can learn from one program to inform another program. So actually, everything we're doing in our genital neuropathy program is actually very informative to some of our other programs from some of the lysosomal storage disorders, for example, some of the other neuromuscular disorders, neurodevelopmental disorders such as Rett syndrome or Angelman syndrome. And you can learn from one to help inform the other. And in fact, we've had in the past 6 months or so, 8 regulatory interactions with the FDA and with other international agencies. And a lot of the comment across a number of our programs, and a lot of the themes that keep coming up are quite common. So I think this idea of learning from one to inform the other and having a more platform approach is a valuable thing. I think secondly, and Mark touched on this earlier, is the value of working with patients and organizations and how we make that real and how we make that meaningful. So for genital neuropathy, and indeed for our other lead programs, we've run workshops specifically where we get into in-depth discussions that can, nowadays we're doing them remotely in the era of COVID and the pandemic, but we have 2 or 3 hour in-depth discussions into understanding from patients and their families what specifically troubles you the most about a particular symptom. What would you like to see an improvement in? And then there's often a very different perspective than the clinicians have. And so we weave all of that, A, into our regulatory interaction, and B, into our clinical development plans. And I'll give you some examples. For example, genital neuropathy, one of the things, this presents often as a motor and a sensory disorder, obviously, but one of the additional features of the disease is a loss of visual acuity over time, which is a very, very sad thing for the patients and families, because these children, as they progress, and they usually die in the late teens or early 20s, these children, as they progress, lose their ability to communicate verbally and orally and physically. And when the vision goes, that's really the last time, really the last real in-depth way the parents have of communicating with their children. And so we made sure to build that endpoint into the clinical trials. And once again, we actually see a stabilization in this loss of visual acuity. So that's an example of something that's more of a humanistic endpoint than a hard clinical endpoint. So this idea of making patient feedback real within the clinical trials is a really important thing. And then the final comment I'll make, and addresses one of the questions, is the question is, could gene therapy possibly reverse axonal degeneration? We've seen this happen for our genital neuropathy study in the preclinical animal studies, where you get a full resolution, a full amelioration in the animal model. I think the key point here, though, and it's why it's important to touch on the diagnostic piece, is that the earlier you treat patients across the whole realm of neurological diseases, the more likely an optimal outcome. Because as the diseases progress, you accumulate disease burden, you're losing neurons. Once you lost a neuron, you don't get it back. So it's really important to diagnose a patient early in the disease process, and treat early in order to stabilize disease then and maximize the chances of recoverability or reversibility, which is one of the reasons programs such as our partnership with GeneDx are really important. I would encourage all the listeners to speak with their clinicians about this.
Well, that's really very helpful. Thank you, Dr. Prasad. And Dr. Foreman, if you want to say a few more words, if you so desire, about Passage and what are some of the important aspects of the company.
Well, I just want to follow up on a few of the things that you just heard Dr. Prasad talk about, because I think making sure that we hear what we call the voice of the patient is so critical. And as you heard, I mean, sometimes the doctors think they know what are the most troubling aspects of the disease. But when you speak with the patients and the caregivers of those patients, you may find out that things are very different. So it's important that we don't lose sight of what we're targeting here. So yes, we want to cure the disease and we want to stop it dead in its tracks and do all of the other associated, and then you get all the associated benefits. But I think we also need to focus on who we're trying to help and what they really is the most troubling. And I've done some work in some other diseases before coming to Passage, where you're almost been quite surprised by what you hear, and so I think that's something I would want to So I'd like we're new to the CMT community. We have the preclinical program that is ongoing. We're hoping that is going to progress well and move into the clinic in the near future. That we're very excited about that. We don't, the time frame for that is hard to say. As you heard, it's hard to predict how preclinical work goes. It's often not a linear path, but so we want to start developing those relationships with the organization, with the CMT2A and the patients that you are serving, so that we're getting the balance right. We're really doing what is needed most by the patients. So maybe I'll stop.
Well, it's really terrific to hear from all three of you how important the partnership with patients has been in the development of both your therapeutics, but also in the design of clinical studies, of course. I mean, the FDA is learning too. And so it was really wonderful to hear from Dr. Prasad, in fact, that you changed the endpoints in your clinical study based on your patient experience. So, with that, I think we're just about arrived at the conclusion of the session. I did want to, however, point out one other panelist that was unavailable to talk today. That's Dr. David Solomon, who's the CEO of Fornext Therapeutics. They have a drug therapeutic that is in an approval study right now. It's a worldwide study. It has shown some promise in earlier stage clinical development that's been published in some peer-reviewed journals, which is exciting to the community. This is a therapeutic for CMT1A that's being explored in adults with mild to moderate symptoms. And there is actually a video that's been posted by Farnext, and I believe that video will be posted on this particular Family Day conference website, so that everyone in the community that is interested in seeing what is, again, a very exciting therapy, please do so. And again, I will say, what it is a delight today to be able to participate in this panel. I certainly want to thank all of the leaders of the companies that are developing therapies. I speak from the community when I tell you how grateful we are that you are really putting your efforts into developing these candidate therapies. And of course, as you well know, you have a patient community that is just starving for it. So thanks so much.
Thank you.
Thanks very much, Tom.
Thanks very much.
Thank you, everybody. Really appreciate you joining. That's really awesome. I just want to share a few pieces of information with the community before we wrap up. But man, this is so exciting to look at this panel. I don't think anything could illustrate the progress we made in the last 10 years. Hearing you guys talking with so much excitement about how you're preparing to do clinical trials on the CMT. So thank you again. And again, for the community, there's a lot more of these companies and more than we can count on the fingers of our hand. And again, when we set up to do this 10 years ago, it was very different. So this being said, the point I want to make, and I'm going to share a couple slides here, if you give me a second. Here we go. So if that works now, this is not the time to let the foot off the pedal. If anything, we need to double down. So what I want to explain to you is why it's so important now to support the CMT program and the STAR program. So I think you've heard today that it's really, and it's recognized as a leading CMT research program. It's drawing great researchers, great companies. But again, we have a lot of work left to do. When I introduced the program, you remember I mentioned that 10 years ago, we were really only in the discovery phase of CMT. And to quote one of our researchers you heard today, Stefan Zuckner, in one of our recent podcasts, he said, well, CMT has entered the age of therapies. And I think it's a wonderful quote. It gives us a lot of hope, gives us a great sense of progress, but we just entered that age, and we need to get through it and get it done. So we have a lot of work left to do. And that's why I think we got to double down on STAR. Now, the other point I want to make is STAR is essentially 100% funded by the CMT community. Basically, except now more and more we're getting support from our partners, but the whole STAR program is funded by the community. So it's your program by you and for you. And this is what makes it so strong. And this is why we have such strong financials, because we actually spend very, very little of your money trying to raise money. Our overhead last year was 9% as defined by Charity Navigator, which is all our administrative costs plus all our costs to raise money. You'll be hard-pressed to find many charities in any field that are that efficient. And again, sure, we do a good job at it, but the main reason is because of you, the community, is because we get all the money and all the support from the community. We don't have to spend tons of money in fundraising and big event and paying superstars and spend marketing money. It's thanks to you, the community. So it is your program, and the reason why it's so efficient is because of you. We got to pick it up. And the point I want to highlight is it's recognized, and all the top rating agencies give us their best rating. I want to say a few words about Charity Navigator because it's the one that's really, frankly, the most difficult and the most detailed and very thorough. And it took us a lot of work to be rated 4-star, which is the maximum. We've been there for a couple of years, and they basically rate you from 0 to 100. And when you're above 90, you're 4 stars. And last year, we were at 99.5. So we were basically almost a perfect score. Now, I can tell you we lost a lot of sleep about how to get there, but now I'm losing sleep about how to stay there. So it's going to be a lot of work to stay there. We'll work very hard on your behalf to stay there. But again, there's probably less than a handful of charities in the whole health sector that are as efficient and turning your contribution into useful programs that we are. And our commitment is to stay there, but we need you guys to continue because we are, again, doing this with very little overhead, okay? The other thing that's very unique about the CMTA is that the board members usually raise at least 20% of every dollar. That means, and why is that important? Of course, it means we have a great board. It also means that the people who make decisions are fully vested. They're voting with their dollars. They believe in the program. They're not just onlookers. They are completely bought in with their own money into the program. So you can trust that those people are going to make the right decisions for you because they're basically voting with their dollars. And the last element, which I think you got kind of a flavor for today, is that when you spend money on some of our programs, it's a bit like being an early investor in a company in the tech sector, like angel fund or early stage fund. But then when we're successful, it brings a lot more money from partners who really basically make an investment be worth 10 or 100 times more. And we've seen that phenomenon more and more, where early investment has been picked up by a company and they write 10 times bigger check than we had in the past. So it really is a multiplying factor for your investment. So in a nutshell, this is very important, is I think why we've been so successful and why we're going to be working on to continue to be successful, thanks to you and thanks to your support. Now it's a journey and we all have a role to play. And I just want to give you a few examples. Obviously, the first one is to support our cause. Money matters. And all what you heard about takes a lot of money. And we're doing really well. I think because of this great support from the community, we actually beat all our records last year through COVID. We have a great year again, but we have to finish the year strong. We have a great fall campaign, so please support us. You can also volunteer, and you see great examples on this picture. There's just wonderful people all across the country, whether it's the youth group, whether it's fundraisers, the branches, the camp leaders, on and on, just wonderful people who do great work and make us be so efficient and so valuable. And also engage with the community. Make sure you get the e-news. We have a very active social media presence, branches. We just launched a new podcast. So there's so many ways that you can stay connected. It can help us. And then join this program called Patient and Partners that you've heard about throughout the whole day. And I think it's very valuable to our partners in the pharmaceutical industry because they want to talk to patients. They want to talk to it. And I think we have thousands of people enrolled now. Last time I looked this year, the number of people even increased by 70% this year. So it's very successful and we need to be more successful. So again, I want to go quickly through those points. I want to thank everybody. Mark your calendars for next year. The next conference will be hopefully in person in Maryland. I'm sure we'll do some sort of virtual conference as well, like we just did. I thought that was very successful. Thanks to the team for that. I want to thank everybody for all your help. I hope you get an impression that everything that's going on your behalf, it's starting to work. There's plenty of work left to do, but we're on it. We need your help. This is your program as much as ours, and let's get it done. So thank you, everybody. And I think the team's going to conclude the day by circling back to the video we showed in the beginning. That's a really nice summary of everything we do and how we do it. Okay, bye, everybody, and talk to you soon. Thank you. Bye-bye.
Thank you.